Hello, everyone, and welcome to the opening of the first International Electronic Conference on Antibiotics, the equal power of antibiotics and antimicrobial resistance. This conference has been organized by MDPI and the MDPI journal Antibiotics, and it is chaired by Professor John Gustafson from Oklahoma State University in the States. I would like to thank our chair and our conference committee for making this conference possible, and all the participants for joining us today. The conference is now open for discussion, so until the 17th of May, you are able to read and comment all submissions presented to the conference. Please check the conference website to find the whole list of submissions, and we encourage you to discuss them. I would like to remind everyone that this webinar is being recorded and that we will post the recording soon at the webinar's webpage, or at the conference webpage, sorry. Today's session is chaired by our conference chair, John Gustafson, and we will have three speakers. Professor Katharina Fromm from University of Fribourg in Switzerland, Professor Eric Brown from McMaster University in Canada, and Professor Helen Zgurskaya from University of Oklahoma in the States. After each speaker presentation, we will hold a Q&A session in which we will address all the questions submitted by live online audience. So please, if you're joining us live, type your question in the Q&A box below and mention the name of the speakers to whom you address your question. Now I'd like to introduce our chair. Dr. John Gustafson is an expert on antimicrobial resistance who presently serves as professor and department head of the Biochemistry and Molecular Biology Department at Oklahoma State University. Over the past 8 years, he has been tasked with improving the department's educational programs and aligning future research programs with the strategic objectives of the Division of Agricultural Sciences and Natural Resources. Dr. Gustafson previously held positions at New Mexico State University in Las Cruces, New Mexico, where he served 10 years on the faculty and 4 years in administrative positions, first as the director of the NMSU graduate program in molecular biology, and then as the associate department head of biology, finally as the biology department head. He also served 2 years on the faculty of the Chicago College of Osteopathic Medicine, and since the beginning of his 24-year academic career at the School of Biomedical Sciences at Curtin University of Technology in Perth, Dr. Gustafson has been successful at securing grant funding to support his research on antimicrobial resistance in bacterial pathogens and recently in science education. So now, Dr. Gustafson, the floor is yours. And to all the participants, please enjoy the webinar.
All right, well, welcome everyone. Good morning from Stillwater and welcome all to the conference, the equal power of antibiotics and antimicrobial resistance. Indeed, I do think that the two of those things do have equal power, but due to the power of evolution, it's my feeling that antimicrobial resistance is taking center stage from these incredibly powerful drugs. So imagine if you were sitting in a hospital bed today and you had stage 4 leukemia or stage 4 melanoma, and suddenly a physician came in and said, listen, we just made a major discovery. We're about to save your life. Or imagine if you were 97 years old and a physician walked into your room and said, listen, we found a new drug that's going to allow you to live for 30 or 40 years more. How wonderful would that be? Well, I get the feeling that Ann Miller, who was 33 years old at the time in 1942 and hospitalized for a month with life-threatening streptococcal sepsis, felt exactly the same way. Certainly her family and physicians did. She was delirious. She had a very high temperature. She was treated with sulfa drugs, which were the only commercially available drugs at the time, blood transfusions and surgery, but none of these had any effect. And then her doctors pulled out this obscure experimental drug called penicillin, which we all know today. From that time on, there was a drop in her temperature overnight, and she was no longer delirious by the next day. Well, she then went on to marry, raise a family, and meet Sir Alexander Fleming, who was the gentleman who actually discovered penicillin. I mean, how is that possible? In fact, half of the penicillin supply was used for this one patient in the United States at the time. She then died in 1999 at the age of 90 years. So I truly believe that Anne and her family probably were able to say that it was a miracle that she was able to get access to this experimental drug at that time in her life. Okay, if we look at the crude death rate for infectious disease in the United States from 1900 to 1996, I like to use this one in my classes. Notice that it started with 40 states that have health departments, and over the years, what happened is the rate of deaths have gone down. During this time, penicillin was introduced, the Salk vaccine was introduced, and then the passage of the Vaccination Assistance Act during this time as well. Numerous antibiotics were developed, numerous vaccines were brought on board, and the miracle of preventing infectious disease from killing human beings had taken off. And also during that time, what happened is life expectancy went up as all of these miracles of modern medical science were coming on board. So as you see a decrease in infectious disease death, it preceded an increase in life expectancy across the world. There was recently a study done on multi-drug bacterial infections between a period of 2012 and 2017. In this study, there were 890 US hospitals that participated, a total of 41.6 million hospitalizations. And they were looking at all these bad boys right here, these multi-drug resistant pathogens, which many of us are very aware of. In 2017, this report provided data to suggest there were 622,000 infections. And of those infections, methicillin-resistant Staph aureus and extended spectrum beta-lactamase producing Enterobacteriaceae infections accounted for 52% and 32% of these infections respectively. And obviously a lot of these are the escape pathogens, once again, many of us are very familiar with. So these two organisms have a massive impact on multi-drug infection across our country. The study actually had some positive findings, which I thought was great. The light blue is community onset infections. The dark blue is hospital onset infections. What they found is that methicillin-resistant Staph aureus infections went down. Vancomycin-resistant enterococcal infections have gone down. Carbapenem-resistant Acinetobacter species have gone down. Multi-drug-resistant Pseudomonas aeruginosa has gone down. And the authors hypothesized that this decrease is probably due to an increased emphasis on infection control in healthcare, including efforts to improve implementation of a wide array of infection control strategies. And we've all seen this. We've all participated in this in one way, shape, or form of another. And so there's an example of where education is working with hospital staff. We need to get that information out to the general public as well. One thing the authors also noticed is that extended spectrum beta-lactamase producing Enterobacteriaceae, there was actually an increase. And note that this increase seems to be more in the community as opposed to the hospital onset. And the authors hypothesized that a greater proportion of cases might have resulted from community-based transition such that healthcare-based interventions would have less effect. Now, there's some people that argue that we're going to witness a post-antibiotic era. And I know that there's a lot wrong with this particular study, but the number is kind of shocking that there'll be 10 million deaths due to antimicrobial resistance by 2050. And these deaths will far exceed the deaths that we see from a variety of maladies in human populations right now. So there, we're proposing that antimicrobial resistance is going to become much more powerful than antibiotics. I mean, also that there will be a massive economic impact due to infections caused by these organisms, which we see right now in terms of hospital costs and whatnot regarding antimicrobial resistance. If you look at this timeline, this is the beautiful golden age of antibiotic development, of something out of one of Silver's papers. And as you can see here, all of these various antibiotics and antibiotic classes were coming on board. This was incredible, so much so that I think around the 1970s, President Nixon actually declared that infectious disease was over, and now we should try to cure cancer. And of course, that was not the case because of the fact that Nixon probably didn't understand the power of evolution. And then we had this discovery void where big pharma started pulling out of producing antibiotics because the economics weren't there for these one-off type drugs that cure a patient immediately. So right now, from this little Pew article that I read before this conference, there's probably 43 antibiotics that are in development. However, as these drugs are approved, resistance will likely emerge. In December 2020, there were 28 that were in phase 1 or phase 2 clinical trials, 3 were in phase 3, and 2 have had new drug applications submitted. Historically, about 60% of phase 3 drugs are generally approved. Within these antibiotics, about 19 of them in development have the potential to treat infection caused by these escape pathogens, these scourge of hospitals worldwide, but also now the community. And the World Health Organization considers these organisms to be critical threats, resistant or nearly resistant to all antibiotics available for treatment. So the power of antibiotics needs to be built up in order to prevent infections caused by these organisms, or excuse me, to cure infections caused by these organisms. Of the 38 companies with antibiotics in clinical development, only 2 rank in the top 50 pharmaceutical companies by sales. Most of these companies do not even have a product on market, to be honest with you. And it's interesting because the large pharmaceutical firms once dominated the field of antimicrobial development. And so this little issue brief concludes that there's too few drugs that are in development to meet future needs. Once again, emphasizing that the power of antimicrobial resistance may overtake that of antibiotics. I love this antibiotic, tixobactin. It's a new antibiotic. Kim Lewis is going to give a talk. He's the one who discovered it in this conference. It has a unique mechanism of action. It actually targets the lipid molecules involved with cell wall biosynthesis and thereby kills the bacteria. But what I really love about this drug is Kim says that it's resistant to resistance. So you cannot select for bacteria to become resistant to this drug. I think that that's a great way forward because literally we'd be able to have some effect on evolution. But the biggest thing about how this drug was discovered was the fact that they developed this ingenious semipermeable membrane sandwich to grow single soil bacterium. If you remember, the vast majority of soil bacteria will not grow in laboratory media. And so they could grow in their soil, in their natural environment, and then express these antibiotics. However, tixobactin is not yet clinically available, but just a wonderful idea and a wonderful technique and just an incredible effort. We all know what causes antibiotic resistance: overprescribing antibiotics, patients not finishing their treatment, overuse of antibiotics in livestock and fish farming, poor infection control in hospitals and clinics, and then lack of hygiene and poor sanitation, and a lack of new antibiotics being developed. But what's really going on here is we're selecting from populations of existing pathogens, microbes that are resistant to the drugs that we use to treat infections. Policymakers have to step up. They need national action plans on antimicrobial resistance. They need to improve surveillance. They need to improve infection prevention and regulate and promote the appropriate use of these medicines. And they need to make information on the impact of antimicrobial resistance readily available, especially to the general public. Nothing good is coming out of the COVID-19 pandemic, needless to say. However, this was a New York Times article, which drew my interest, is that the confidence in medical scientists to act in the best interests of the public is going up, as well as the confidence in scientists to act in the best interests of the public. I still think that the quantity of people who think we're doing a great deal to work in the best interests of the public is something we should all be very concerned about. And we need to get our messages out to the general public, not just to our colleagues and the scientists at large. I think that's critically important. So I guess one of my major take-home messages is always take time to educate the general public on antibiotic resistance. Have your 3-minute elevator speech always at the ready to discuss what you're doing in your laboratories, why this is important, and more importantly, that we have extant evolution that's occurring right in front of our eyes with antimicrobial resistance. Literally, in a sense, it's evolution that we're attempting to fight against. And with that, I'd like to throw out a little bit about Antibiotics, the journal. We're going to have a special antibiotics issue. The deadline will be September 30th, 2021. You will be able to get a discount. The impact factor on antibiotics has gone up, 3.893. And then the turnaround process for peer review is very short. And at the same time, reviewers can get recognized with vouchers that entitle them to a discount on MDPI publications. And of course, this is an open access journal, which is free to the general public. And with that, my thanks goes out to the conference committee, all the speakers and presenters, and the wonderful MDPI staff. And I hope all of us continue to support government funding for basic and curiosity-driven research. Thank you very much. And now I'd like to introduce Katharina Fromm. She is a full professor in the Department of Chemistry at the University of Fribourg. She's also the Vice Rector of Research and Innovation at the University of Fribourg as well. Apparently, you just started this job in 2020. I bet you that's a lot of extra busy work that you have to deal with. She did her postdoctoral work at the University of Tübingen in Deutschland, obviously, with Professor Jean-Marie Lay, who was a Nobel Prize winner, and also at the University of Strasbourg in France, and also at the University of Karlsruhe. She then went on and became a junior group leader at the University of Geneva, and then moved on to the University of Karlsruhe, and then was in the Department of Chemistry at the University of Basel in Switzerland, and now she is in her position in Fribourg. The title of Katharina's talk is going to be, Can Bacteria Become Resistant to Silver? New Insights into Bioinorganic Chemistry of Silver. I'm very excited to see your talk.
Well, thank you so much, John, and welcome everybody to this talk. It's a great opportunity to present our latest research here. So I will speak about silver in this afternoon's talk. And this is one of my research areas in the research group, among others. So we look into nanoparticles, nanocontainers for antimicrobial applications, but we also do mixed metal compounds for oxide materials, batteries, and so on, stimuli responsive particles or polymers, coordination polymers for antimicrobial coatings, anthracene-derived ligands for optical sensing, and then this will be the subject of today's talk. This is about bioinorganic chemistry of silver, which is of course related to some of the others as well. If we think about our human body and the many pieces that we can replace, there are a lot of body foreign materials that can be inserted into the human body. And here's kind of the maximum of implants kind of that you can get overall in such a human body. And all of these body foreign materials, they have one thing in common. They do not have a natural defense system as our natural bone has. And therefore, all of these implants, they are prone actually to bacterial adhesion to their surface and therefore also to getting infected. So if we look a little bit at one example of such an implant, namely the hip implant or hip replacement, you can see that while the international average is around 166 persons per 100,000 in population that would get such a hip implant, the developed countries have much higher numbers, and Switzerland is among the top of those who actually use hip replacement to make life easier. So as I mentioned before, these artificial materials, they can get infected. And there have been, as John mentioned already, a lot of antibiotics that have been actually discovered roughly in the 20s, 30s, 40s, and so on, and that were used over a wide range, time range here until today. And we've seen that also there's this resistance problem that occurs with these classical antibiotics. Now, if we look at silver as an antimicrobial compound, we can actually see that it has been used for ages already to treat, let's say, or to prevent bacterial infections or to treat ulcers, but without knowing very much about what was going on. It was just the fact that, for instance, drinking water stayed fresh in silver vessels much longer than in any other containers. So in about the 90s or so, people rediscovered the silver as the superbugs emerged. And now there has been quite an intense research done on how silver can act as an antibiotic and how this is happening at the molecular level. So there wasn't much known because actually during all these times that many of these antibiotics were used, there was not much research on silver at the same time. So in my group, we have been developing silver coordination compounds, and where silver is bound by a ligand to the surface and then connected by other ligands, which can be antibiotics, for instance, like vancomycin, to other silver ions to form a coating on the surface of an implant. And you can see here how this coating is presents itself under the electron microscope. So it's a nanoscale coating, which is hardly visible by the naked eye. And if you compare now a coated versus an uncoated model piece of implant, which can be, for instance, titanium, you can see that the coated sample has a much larger inhibition zone around this implant piece as compared to the uncoated, where the bacteria grow everywhere. So this is shown in a different light here. And these kind of coatings, which are based on ionic silver, they are nanostructured and they are antimicrobial in vitro, as shown here, and they can last up to 3 months approximately. We have tested this also on dental implants, so dental coatings that were tested in situ in a flow chamber. Here is this flow chamber. Here we have a tank with saliva taken from several people, then sterilized, and then we added to this one of these early colonizing bacteria as sanguinis, and we looked at what happens after, let's say, a certain time that we let this saliva flow over these samples. And you can see that in comparison, uncoated versus coated samples show alive bacteria on the surface, sticking to the surface, whereas the treated, so the coated surfaces, they have only dead bacteria sticking to the surface of the titanium or gold plates. So silver, our silver coatings, they seem to be quite efficient in killing the bacteria near the surface. We next went into a mouse model, and here you can see a mouse that has inserted here little cages made from titanium or polymer that get inserted under the skin. And these cages in our experiments were titanium cages, respectively the silver-coated titanium cages. And we could show that in terms of leukocyte viability, this worked out quite nicely. It reduced a relatively strong infection of S. epidermidis. You can see this reduction right here. And after 14 days, you can see here that even in vivo, we still have remaining silver that is being released. So it's still active to protect the implant from bacterial adhesion. So all of this works, but rather on, let's say, weeks of action, like 2 weeks to 2 months, let's say. But of course, if you have a hip, an artificial hip, you have to have longer release of silver in order to prevent bacterial adhesion to such implants. And so we have also developed in our lab nano containers that can have a drug inside or can be soaked to have a drug inside or a silver particle that can be either inserted as a core shell particle or as a nano rattle structure where there is a void between the nanoparticle and the outer shell, or else we can embed these silver nanoparticles in the shell itself. With a double-coated shell, as shown here, we have 2 coatings, silica coating and titania coating or ceria coatings. Then we can actually prolong the silver ion release over many, many months. And we could show that in this particular case of a coated implant, after 3 months roughly, we had only 10% of the total silver that was loaded that was released. So you can extrapolate that actually this kind of coating could last up to 30 months, which is, I think, one of the longest active coatings that I know of. So how does silver actually function? So if you go to the literature, and there is quite a number of that on silver, it is said that silver compounds can damage bacteria in many different ways. One is by breaking already the cell membrane. Then it will inhibit the function of proteins and also protein expression. It can bind to the DNA by binding to A and G. And it is forming directly or indirectly also reactive oxygen species. And all of that together will inhibit cell respiration and the bacteria will die. So this is how it is said to function. But whenever I explain this to potential users of these silver coatings for implants, there comes the question, yes, but what about resistance against silver? Can bacteria become resistant? Well, resistant as against antibiotics, probably it's not the same way, but there are ways that bacteria can cope with high concentrations of silver by using efflux pumps. That's one way. And another way is by reducing ionic silver ions to silver nanoparticles. This second part I will not be able to cover today because it will take much longer. So I will speak mainly about the efflux pump mechanisms. So in the EAT, there are bacteria, gram-negative bacteria, that can survive in relatively high concentrations of silver. And they have a resistance mechanism that is based on this efflux pump. And there are two possibilities actually to work with an efflux pump. One is the copper efflux system, the CUS system, which is then usually combined with a loss of outer membrane porins. And there is a second efflux system that is the silver efflux pump, which has something special, namely the expression of the CE protein. So if you compare the copper and the silver efflux system against each other, you can see that the silver has a couple of more proteins here, and in particular the CE protein, which is unique to this particular efflux pump. The copper efflux system does not have this protein, but it has also the QSF protein, which is similar to the CLF protein, and it is able to bind a single silver ion and transport the silver ion then to the ABC or CL-C, CL-B, CL-A complex here that is responsible for transporting out the silver ions. But the role of the CL-E was unknown. So if we look at the interplay of all the proteins, we start with CL-S and CL-R. In the presence of silver, they start to produce all the other proteins, and then silver gets bound by CL-P, exported into the periplasm, bound by CL-F, which transports it to the CBA complex, and then there is a chemiosmotic silver proton antiporter that takes action. For the CL-E, there was a big question mark, what would be the role of this? And Simon Silver, as the name nicely says, has worked on that actually to start with. So he proposed, or he analyzed this protein, the CL-E protein by NMR, and found that it had quite a high amount of histidine. And in his studies, found that the protein, the CL-E protein, could bind up to 5 silver ions, and at the same time go from an unstructured protein to an alpha helical protein. So that was actually the starting point where we got interested in this CL-E protein. And what we ask ourselves, so what is the structure of the holo CL-E, and what is the mode of action to deal with these silver ions? My student Valentin Chabert at the time, he actually looked a little bit closer into the sequence of the CL-E protein, which has roughly 143 amino acids, and found that next to the histidine moieties, there were also methionine amino acids, and this usually either to form like a tripeptide or tetrapeptide entities in this CL-E protein. And you can see here the different motifs that we have identified, and we wanted therefore to know whether methionine was actually involved in the binding of silver here. So we first used methionine and did an NMR titration with silver nitrate, and yes, we could confirm that it binds to the thioether chain. We were then lucky to obtain a single crystal structure, which is actually forming a coordination polymer in which the silver ions are bound by 2 silver and 1 oxygen atom of 3 different methionine amino acid moieties. It forms a coordination polymer actually. So yes, we could confirm that methionine contributes to the binding in CL-E, and we next did a titration of such a of these tetrapeptides and tripeptides that we had in the sequence. So we synthesized them first, and then did NMR titrations with these tetrapeptides. And here's just one example, HEFM, which we titrated with up to 2 equivalents of silver. And you can see nicely that after the addition of 1 equivalent, there is no more shift in the NMR signals. And so that we can actually nicely see here that obviously a 1-to-1 complex is formed, both involving methionine and histidine. So this was the result from our NMR studies, and the complexation is 1-to-1 for all of these tetra and tripeptides that I have just shown you before. We then determined actually by a competition experiment with imidazole, the binding affinities for the different binding sites as they are shown here. And we can see that these values are between 5.3 and 6.4. Respectively 6.6 for these tetrapeptides and tripeptide that we have. So obviously there seems to be a silver sponge effect, and silver can bind, the CLE can bind, at least in our hands, up to 10 silver ions, 9 silver ions. The structuring effect of these tetrapeptides, we could not fully confirm that it would form alpha helixes because simply these pieces of peptide are too short. So from the CD spectra, we could just see an indication, yes, there might be alpha helix formation. So at that time point, actually, there was another paper appearing by Asiani et al. About the CLE protein, where they state that there are specific binding sites where the silver binding occurs in a cooperative manner. And so this caught our interest. And since we only had currently looked at individual binding sites, we wanted now to look at larger excerpts from the CLE protein. So we made these longer peptides here that had now 2 binding sites, and that were here actually linked either by 6 amino acids that were the A peptides, which we synthesized here. So there are 6 amino acids between the 2 binding sites. Or we also made the B1 and B2 peptides, where we have only 3 amino acids between the 2 binding sites that we had identified. So we now took these longer, synthesized those longer peptides and looked again at the binding properties. Interestingly, and this is of course something to be careful with, if you do just if you just take the sequence, the amino acid sequence that we have in these 4 peptides, and you do a structure prediction, they already predict a helical structure. However, if we look at NMR data or at also CD spectra, so this is, sorry, first start with the NMR data and NOSI spectra, we do not see an alpha helix formation. The CD data will come later. So again, we did further silver ion titration experiments, and we could show that for all of those peptides, we have actually 2 equivalents of silver ions that can be bound, after which there is no more NMR shifts to be seen. So if you go to higher quantities of silver, there's no further effect. So again, we can say that, yes, there are 2 silver ions bound on these longer peptides, and now come the CD spectra, where we can see that before addition of silver, there is a relatively unstructured peptide, whereas upon addition of silver, we clearly start to see, and this is very strong for the B1 and B2 peptides, we really start to see the alpha helix forming. So it becomes clear that the theoretical prediction should be taken with care. There was no helix before addition of the silver here. So modeling now from the NMR titration structures, we can see that indeed A1 and A2 with the longer spacer in between, they are not so nicely structured, but indeed the B1 and the B2 peptides with a short spacer in between the 2 binding sites, they really form nice alpha helices. And you can see this would be then the places where the silver ions bind. the histidine and the methionine, they nicely point towards each other. And this is exactly how the silver can fit in. So we now determined the binding affinities for these binding sites by actually using a fluorescent titration of a tetrapeptide fluorescent probe as a competitor to our peptides. And we look at the quenching of the signal as we add then silver and the longer peptides that we have. And from there, we could then actually determine the different binding affinities. And what comes out in the end is this table here for B1 and B2, A1 and A2. Here are again the individual binding motifs, these tetrapeptidic entities that bind the silver. And we could see that actually in terms of, so these are the binding constants for the short peptides, and these are the binding affinities for the long peptides. The first binding event in our hands for these peptides was always the strongest one, and the second one was usually weaker. So in our hands, with these peptides that we have here, we could not observe a cooperative effect as it was shown for the long protein by Asiani et al. But that doesn't mean anything. I mean, these are just some excerpts from the long protein. And of course, there are other combinations to be studied. So what can we now say about the CLE? We have for the CLE found binding affinities between 5.3 and 6.6. We, from the literature, know that CLB has also binding sites, 2 binding sites at least, from which is known today, with a binding affinity of 7.1 and 7.6. So if you look at that difference, it should be in principle possible that CLE, after binding silver ions, could transfer its silver ions to CLB, which then is responsible for the silver efflux. At low concentrations, however, this is probably not necessary that the CLE binds these silver ions. However, as the silver ion concentration increases, the CLB sites might get blocked, and CLE can actually act as a sponge for these larger numbers of silver ions. And this becomes really, really important if we go to even higher concentrations, where then the silver is indeed caught by the CLE before being transported out of the cell. So in order to look a little bit closer into this particular theory that we have now from the binding constants alone, we actually asked the question, is silver only a chaperone, a sponge, or is it also a chaperone? So can it do both? And for that, we actually started again another collaboration. So we are in close touch with Maggie Ologne from Lyon, where we now looked into CLB. And here you see the sequence of CLB. So it's a much longer protein, and an analogon of CLB at the N terminus shows quite nicely here that there are a couple of methionine groups that can bind the silver ion. This is also from an NMR structure. And if you look at this the sequence here, which is just increased a little bit so that you can read it better here, we do have indeed quite an impressive amount of methionine, 4 of them, but we also have histidine there, here, and here at the very end. And so we decided to synthesize this peptide, this long peptide here, for which we did NMR titration experiments with silver ions. And we found that this piece of the CLB protein was actually able to bind 2 silver ions. And this first in a, let's say, normal manner from what we expect from our binding affinities. This is a little bit lower than what we know from histidine-methionine combinations. So this is rather a methionine-methionine combination that is involved here. But, and this is now different from what we have observed so far in the CLE peptides that we made, the second binding event seems to be cooperative. And this one involves methionine and a histidine. So this is interesting here that we have now here a very, very strong binding for the second silver metal ion. The question now, let's look again at the CLE, and there we chose for this particular study again the B1 that was known to form a very strong helix upon the binding of silver ions, and which had these affinities here 6.2 and 5.2 for log K1 and log K2. So it was not cooperative. And with those values now in hand, we thought, okay, both proteins or both peptides can bind silver, but can they maybe hand then the silver ions to each other? So is there one way or another where this exchange can take place? And so we put actually together those different combinations as follow. In the first experiment, we loaded the CLB peptide with silver and put it in solution with the silver-free B1 peptide of CLE. In this case, we don't see any change at all. So this means that the CLB peptide will not hand over silver ions to CLE, to the free CLE B1 peptide. So that was the first experiment. And then we did another experiment where we had loaded CLE B1 peptide with 2 silver ions, which we put into solution with silver-free CLB peptide. And there we saw that indeed the 2 silver ions from CLE B1 were transferred to the CLB peptide, which had no silver when starting the experiment. So we see a clear shift here in the NMR titrations. And then we did another experiment. We looked at a mixture of silver-free B1 from CLE and silver-free CLB peptide. And then we added silver ions. We added this in a titration experiment, and we could see that indeed then the CLB peptide binds these silver ions first, and only at the higher concentrations, then also the CLE, the B1 starts to bind. So this shows already a little bit the functioning of those 2 proteins together. So as long as the silver ion concentration is low, then CLB can cope with the silver concentrations. Whereas if we have a higher concentration of silver ions, then also the CLEB1 peptide in our hands starts to bind the silver ions. And that would actually reflect a little bit what we have already hypothesized, namely that the CLE acts mainly as a sponge at high silver ion concentrations. And indeed, if we now lower the concentration of the silver ions, then also the B1 CLE, which is loaded with silver ions, in contact with the CLB peptide, will then transfer the silver ions to the CLB peptide. So overall, the CLE B1 binds the excess silver ions so that the CLB peptide can function. And in this sense, of course, the CLE B1 is rather to be seen as a sponge than a chaperone. However, as said before, once that the CLB binding sites are free, then at high concentration CLE is loaded, the CLE will nevertheless give its silver ions to the CLB peptide. Of course, we've done this now with short peptides, and this was only done with B1 for the moment, which is said to be one of those pieces of CLE that binds in a cooperative way in the protein. And therefore we have to verify this, of course, with the entire proteins. And we are currently doing this in the labs, my labs, and also in Lyon together, so that we can study these interactions in more detail. With this, I come to the end of my talk, so we have some time for questions. But before giving you the floor for these questions, I have to thank a couple of people here. Michael Fuß actually helped us with the bacteria that actually can deal with silver outside and that are reducing. Lucille did the work that I just presented you at the end. Valentin worked on the tetrapeptides and the A1, A2, B1, B2 peptides. Bernd Giese is my visiting professor, permanent visiting professor and husband at the same time, whom I thank also for fruitful discussions. And today we have Max in my group, Maxime Karamash, who is currently working on the second part of my talk, which I could not present today usually, which is about how bacteria can reduce silver ions outside of their cell. So for the collaborations, I have to thank Maggie Ologne and Olivier Senek and Olivier Walker, as well as Carlos Salguero in Lisbon, and of course, all my funders and supporters and the rest of my group, who is just fantastic, even in COVID times. So thanks a lot to all of those, and thank you also behind the screens for your kind attention. And of course, I'm happy to take any questions.
Okay, we have a question in the chat. Is this SIL ag resistance system specific to silver only? And have you tested other translation metals? And how would you comment on the specificity of this system?
Okay, so obviously the SIL-E system has so far only be found where silver was really, where bacteria were exposed to silver. That means in nature, where we can find them, for instance, in silver mines, where there is a lot of ionic silver around. That was in nature. And in the medical application, you may know that silver wound dressings are usually used a lot in for burn wounds. So if you have large skin areas that got burned, you usually work with silver-coated wound dressings. And it was found that under these wound dressings, especially if they have to stay on for longer times on such a burn wound, then a bacteria obviously can catch a how do you call again the spherical DNA, the plasmids, and can actually express the silver efflux pump system. So the copper efflux pump system is known to be able to pump out silver ions as well. And although I have never tried other metal ions as copper, I would guess that the silver efflux pump would also be able to pump out other metal ions, be it copper or mercury maybe as well, because they have similar binding sites. However, if then SIL-E has exactly the same role, this remains to be seen, because silver has, of course, a specific coordination. It has a linear coordination. So coordination number 2 for such a big metal ion is not that frequent. So other transition metal ions wouldn't prefer to have a linear coordination, but there are, of course, gold could be one, and mercury also likes linear coordination. So there might be other metal ions that can be exported by the SIL system to be tested, to be honest. But it's a good idea to test that. Thanks.
That question was from Jitender Mela. I hope I didn't butcher your name there, Jitender. Also, it wasn't translation metals, it was transition metals. Thank you, Jitender. We have a question from Anthony Coleman. Is it not that eukaryotes are resistant rather than prokaryotes?
Yeah, so it's true that silver is surprisingly harmless, let's say, to our own cells. And there is always the question, why is that so? And again, this is likely to a different composition. So first of all, we have in our cells, we have the nucleus where the DNA is typically protected, whereas bacteria don't have a nucleus. So the nucleus, the DNA is easily attacked in bacteria. So that's one thing. But on the other hand, we also have other proteins, respectively more of certain proteins that are responsible for heavy metal sequestration or capture, let's say, so that it is for us easier to bind the silver and to kind of make it, useless or inactive. So it can harm us, but before it harms our cells, it will first kill the bacteria. So there's a therapeutic window. And it is important for that to actually control very well the silver ion release. That is important not to have a too strong release. And if you can tune the silver ion release, we have in our experiments shown that it can be biocompatible on one hand and it can be antimicrobial on the other hand. So that is, of course, a big challenge because the systems are very complex. We are in biological fluids. There is chloride present. There is a lot of different compositions that you can have, and handling silver ions in general in biological media is tricky, and you have to be very, very careful when you do these experiments with bacteria or with cells in general. If you have chloride and phosphate and so on, you need to take this into account and be very sure about what you really have in solution and which effect do you really measure.
Professor Fromm, we have another question from Hai-Sun Cho, who thanks you for your wonderful presentation. I'm sure we all thank you for that. In terms of therapeutic application, do silver particles cause cytotoxicity if they use them along with antibiotics?
Okay, so what we have shown in our own studies is that using silver in combination with antibiotics made a synergic effect on bacteria, so that the two together are better than one of each alone, or just by taking the sum of them, they are stronger than the sum, than the simple sum. So that's one thing. Now, the other question concerns what happens to the silver nanoparticles in the body. So we have studied a lot of our nanораttles where we actually wrap the silver nanoparticles into, for instance, a silica shell or a titania shell, to be able to control the silver ion release. And what we observed in a recent publication, which I can share if that's a desire, you can send me an email about that, that is then actually that these silver nanoparticles can be taken up by cells like macrophages. And we can show nicely that they will stay actually in a compartment even after longer times. And we could not see any decrease in the alive cells of the macrophages. We could not see getting less active, or we could not see any inflammation effects. So this is something where we are quite optimistic that silver can, to a certain doses, be used also in the body. And let me say this with the Paracelsus. So everything is a toxin. It just depends on the doses. So in the eat, on the quantities. So we have this, of course, with silver as well. And you may know that people who get exposed to a lot of silver, or who exaggerate taking nano silver because it's healthy and they have their nano silver drink every day, they can actually develop a certain condition which is called argyria, where the skin can turn gray, and where people get very light sensitive. However, these people can live with argyria, but of course there might be long-term effects like in the kidneys and so on. So overdose of silver nanoparticles should be avoided. But depending on the quantities, I guess it's controllable.
It sounded like silver can turn people into vampires there, Professor Fromm.
Kind of, yeah.
Just
Although
a joke, everyone.
silver should kill the vampires, right?
Yeah, correct. Kevin Schindler has a question. Can octahedral metal transition base complexes do the same job with extra ligands? And then he has in parentheses antimicrobial active. I think ligands is what he's talking about.
I see. Yes, yes. So there have been studies done where antimicrobials have been coupled to, antibiotics have been coupled to other metal ions as well. And so, I mean, there are even iron compounds that can be antimicrobial. Copper, zinc is our antimicrobial metal ions. So yes, depending on which ligands you give them, you can use them also as antimicrobial drugs, absolutely. So there is little research done on larger ones like bismuth or gallium, which are also antimicrobial. But I think there's a lot of potential to look into those as well. The question is always how active they are compared to their weight and how much of these metal ions you need to provide to have the antimicrobial activity. And in that sense, silver is one of the strongest antimicrobial metal ions that we know. And the problem with copper and zinc is that these are bioactive metal ions. So we have in our body zinc and copper ions, and therefore this can very quickly actually interfere with our own homeostasis of these metal ions, whereas silver doesn't have a role in our body. So there we have, it is a body foreign element, if you wish, and so the body can interact with it, but it doesn't immediately interfere as does copper or zinc.
And Professor Fromm, one last question for me. We got a minute. Does mutation play any role in metal resistance in bacteria? Have there been laboratory studies done on that?
Right. So we looked at a couple of mutations in the other part of which I couldn't talk much about, but where we use anaerobic bacteria that are able to reduce metal ions outside of the bacterial cell. So in the sense of sensitivities to silver ions, these bacteria can cope with silver ions simply by reducing these silver ions to silver nanoparticle outside. That means that the bacteria have to transport electrons from the inner membrane through the periplasm to the outer membrane. And this happens with cytochromes. And there are bacteria such as Geobacter sulfuretocens or Shevanella, which do have a large amount of those cytochromes in the periplasm and at the inner and outer membrane. And when you do mutations, when you do studies about these effects, you can see that some cytochromes are more important than others to do this metal ion reduction. With respect to the silver efflux pump, there have been mutations where the CLE was actually deleted. And there it was shown that the CLE was essential for the functioning and survival of the bacteria in high levels of silver ion concentrations.
Wonderful. Thank you so much, Professor Fromm. A round of applause on the internet for Professor
Thank you.
Thank you so much. And with that, we're going to move on to the next speaker, Dr. Eric Brown. Dr. Brown, if you could please share your screen. Dr. Aaron Brown is a distinguished university professor in the Department of Biochemistry and Biomedical Sciences and a member of the M.G. de Groot Institute for Infectious Disease Research at McMaster University. Dr. Brown is a fellow of the American Academy of Microbiology and has received a number of other awards, including the Canadian Society of Microbiologists Murray Award for Career Achievement, and the Canadian Society for Molecular Biosciences Merck Frost Prize for New Investigator. He currently holds the Killam Research Fellowship from the Canadian Council for the Arts and a Canadian Research Chair in Microbial Chemical Biology. His talk today is going to be on a systems approach to antibiotic drug discovery. Dr. Brown, take it away.
Thanks so much, John. Real pleasure to be here to tell you about some of the work that's going on in my lab. I'm going to focus for today on our efforts to use systems approaches to discover new and interesting compounds that may have value as leads for antibiotics. John showed a kind of a version of this slide earlier, and I want to revisit it to make a fresh point here. This is, of course, work again that Lynn Silver did some years ago to sort of get a sense of when antibiotics have been discovered. This is the date of their discovery, and they're binned on the decade as new antibiotic classes, unique chemical classes and mechanisms. And it's actually kind of a refreshing look at it by the folks from the Pew Foundation. There's the golden age of antibiotic drug discovery, right, where most of our molecules come from. And over here, this visual makes a very good visual for the lack of discovery that's happened in the last 30 years or more. This is a fairly depressing thing for a biochemist like myself. I grew up during this era. Recombinant DNA technology comes to the fore in the 1980s, and things like high throughput peptide synthesis. You have advances in robotics and liquid handling, structural biology, genomics, all the sort of stuff again that was really formative science. For me as I kind of grew up as a biomedical science in this space. And while it's been extremely successful in yielding drugs for other therapeutic areas, it has been an utter failure in the area of antibiotic drug discovery. And so that's in stark contrast, of course, to the methods that were used in the discovery of drugs during the so-called golden age, where it's a remarkable therapeutic area where we have fantastic in vitro assays like this dish. If we can get those to work in a mouse, it's really predictive of what goes on in a human. That too is unlike almost any other therapeutic area where the mouse is a great predictor for what goes on in the human. And so we've kind of got to thinking in my lab that the vast majority of antibiotics have been discovered with this age-old paradigm, and so is there something that we can learn from this? And so really it's a question of, have drug discovery approaches that are reductionist in nature, such as those that are used in modern antibiotic and the discovery of other drugs, are they failing for antibiotic drug discovery because we don't understand the system as a whole? And so can systems biology then offer some unique insights into the discovery of new antibiotics? And so I'll talk, there's really a wide variety of systems approaches, and I'll talk a lot about these two tool sets today. So just to introduce them, the KO collection out of the NAR Institute in Japan, Hirotada Mori's lab, where many years ago he made deletions in all of the genes that could be deleted on rich microbiological media. And the Alone collection, also some years ago, created in the laboratory of Yuri Alone in Israel, where each of E. coli's promoter units was fused to fast-folding green fluorescent protein as a reporter on those promoters. And so, we've been interested in my group in using tools like this in a systems mode. The idea here is to chart genetic interactions systematically on a genome scale. We're talking about the interaction of genes with other genes, the interaction of genes with chemicals, and all the while we're using formal genetic principles of enhancement or suppression of phenotypes. And of course, most commonly, the phenotype associated with antibiotics is that of growth inhibition. So if we look here, first off at gene-gene interactions, we use what has been kind of popularized by the the yeast field, the so-called multiplicative rule for establishing an interaction between genes. So we have growth on the Y here, and we're looking at the growth, say, of wild type, indexing that to 1. You delete gene X, you get a 70% growth rate, or a 30% decrease in growth rate. Gene Y may have a growth rate, say, of 80% that of wild type. And then the idea, of course, is what is the expected growth rate for a double deletion where we've deleted both gene X and Y. The multiplicative rule puts that expected growth rate at 0.56, the product of those two deletions. And if it grows worse than that, we call that enhancement. That's often called synthetic sick or lethal. So it's an enhancement of the growth inhibitory phenotype or suppression of that phenotype. And of course, we can do the same thing for chemical genetic interactions. Here again, we have gene X growing, right, with a growth rate of 70% of wild type. And if we put chemical number 1 in it, some concentration, perhaps this is a sub-inhibitory concentration of an antibiotic, it's now growing at 80% of the growth of wild type. Again, these two together in a tube, we expect a growth rate of 0.56. Less than that, we call that enhancement. Greater than that, suppression. So let's talk first about chemical gene interactions. Here again, we're focused on this collection of deletions out of Mori's lab in Japan, some 4,000 deletions or so in E. coli genes. Key to this work is really having, great robotics and liquid handling, but also colony array equipment. Shown here is the Singer rotor that we use quite a bit. And this is a 6,144 density array of the KO collection on a slab of LB agar, right, which is uniwell, microwell size in dimensions. And so you can see that there's really great separation of colonies. We've got an edge effect that we can deal with various approaches, but in all, it's a really remarkable ability to collect data on the growth of a large number of clones in a small space. So enter Sean French in my group, whose claim to fame, I think, in this area is this idea of trying to take a dynamic analysis to measuring the growth of high-density arrays of colonies like this on solid media, in order ultimately to build mechanistic fingerprints, right, where we can try to make some conclusions, generate hypotheses, and really just better understand the action of known antibiotics and try to understand the action of new ones. And so this approach has used the following kind of approach, where we have transmissive scanners that are in an incubator. We can put 4 such plates onto any scanner here, and we're watching the absorption of light or the transmission of light through the sample over time as it's growing. And shown here is not a simulation. This is a single colony, right, growing. We're of course sped up over time, and we're looking at the integrated density that we are recording using these transmission scanners of this colony. And you can see we get a really great view of a growing colony in this high density array. And so we're a little bit compulsive about this stuff in my lab, I confess. And so at some point, one of the group members, really cut out all of these colonies and recorded the growth, the integrated density as a function of colony forming units, and it turned out to be a straight line. And what that meant, of course, is that at very high density now, we could collect the growth rate of any one clone here. I'll add that, of course, most of high density arrays of microbes, most notably yeast, most information is really gathered out at this endpoint, and there's quite a bit of error and sort of issues with collection of growth at that endpoint that are resolved really at collecting kinetic data, where we can get, for example, a doubling time, right, from the early growth phases of this colony that is characteristic of any strain. It's relatively independent of the inoculum that goes onto that plate, and it's really quite a bulletproof parameter in understanding the growth of that particular colony. And so, here's a look at, it's the requisite systems diagram network of the interaction of some 15 antibiotics with the, with each of those 4,000 genes or so that we have deletions in E. coli. Shown in green here are growth rate data, gray is endpoint data. And it turns out there are some 1,000 interactions if one looks at the endpoint data, about 1,500 interactions if we look at the rate data. And so really, looking in either of that way is remarkable. I mean, we're seeing an incredibly high density of interactions with the rate data of about 100, of course, per chemical. That goes way beyond the proximal targets of these chemicals and gives us a sense of a highly networked and a highly dense network that is buffering the activity of antibiotics. And so I want to tell you how we've used some of this information really to go about our thinking on, for example, the gram-negative challenge. And this idea, of course, another lovely diagram out of the Pew Foundation, where virtually all antibiotics that have been discovered work really well in gram-positive bacteria, but gram-negative bacteria, where we're hearing has the greatest medical need these days, is an area where a relatively small proportion of antibiotics actually work. They're of course subject to the permeability barrier that is the outer membrane, and also to efflux that we heard a fair bit from Katharina, and we'll hear more, I'm sure, from Helen. But suffice it to say that this is a real issue of interest to me in my laboratory as well. We have, as shown here, a large number of molecules that are gram-positive only, from oxazolidinones to glycopeptides to macrolides and rifamycins, that we might wish could be quite effective in the treatment of gram-negatives, but they are not. And so, we've been thinking about, what it might take, for example, to get a molecule like rifampicin or erythromycin to be active against a gram-negative organism. And so shown here is a chemical genomic experiment with both rifampicin and erythromycin. We're asking for genetic enhancers of the action of these antibiotics. And so rifampicin, we've got, what is that, about 138 genes that are leading to enhancement. And if we look at this slice of the Venn diagram and have a look at the enrichments in terms of Go terms in E. coli, we see lipopolysaccharide biosynthetic genes are lighting up very much in the top of this. Erythromycin as well, similar genes, and the intersection of those, other classes of genes that are involved in outer membrane assembly. All of this, of course, points to these pathways for the biogenesis of the outer membrane of E. coli and other organisms involving things like lipopolysaccharide synthesis and transport to the outer membrane, protein synthesis and transport, lipoprotein synthesis and trafficking, and similarly phospholipid synthesis and trafficking to the outer membrane. And so, clearly these are processes which are extremely important, to maintenance of this, to maintaining this outer membrane, but also to processes like efflux that are dependent on the presence of the outer membrane. And so our question was, could we use systems information to try to come up with new screens, new chemical screens for compounds that would target the outer membrane? And so a little serendipity helped us out here. This is John Stokes, currently a postdoc at MIT, and Jim Collins' lab. You were going to hear more about from I think probably John's work as well from Jim later on in the conference. But John made this remarkable discovery in in an eLife paper that was focused on ribosome assembly, in fact, that vancomycin is active against E. coli at low temperatures. So this is 15 degrees, and we're seeing a sub double digit MIC for vancomycin under these conditions, 15, 20, 25, 30, 37, and 42. So this temperature dependence of vancomycin was of great interest to John. He went looking for genetic suppressors of that sensitivity and discovered that in a screen of the KO collection at 15 degrees that there were several mutants, in fact, that led to the suppression of this growth inhibitory activity of vancomycin at those temperatures. Go classification of those processes highlighted the very processes that we saw earlier were important, of course, to outer membrane synthesis. And at that point, a light went off for both John and I that we had a very interesting opportunity to do an antagonism screen for new compounds that would impinge on these processes, where very slow growth at 15 degrees in the presence of vancomycin would lead to cell death, but that a chemical that would act on these processes would give rise to a growing colony. So we have a screen for life, a great target list, idiosyncratic genetics that gives rise to a very low frequency of active compounds, little or no nuisance compounds from a screen like this, and increased sensitivity over a screen, for example, where we would just look for potentiation of the action of a gram-positive only compound. Ironically, perhaps, John didn't pull out an inhibitor of one of these targets. He pulled out a compound that actually impinged on the outer membrane directly. And that compound was pentamidine. This was a pilot screen of off-patent drugs. Pentamidine is a molecule that's been used for some time to treat fungal pneumonias. It's an antifungal molecule with this characteristic diamidine structure separated by these 5-methylene groups, hence the name pentamidine. And so it turns out, as I alluded to, that pentamidine, in fact, is a physical chemical perturbant of the outer membrane of E. coli. And this is atomic force microscopy, where we're looking at a probe being dragged across the surface of E. coli, and what we see for wild type are nanometer modulations in the cell surface treated with pentamidine, and we see these deep furrows that are associated with a disturbance, a perturbation in that outer membrane that is reminiscent, of course, to the sort of cell surface that we see on treatment with a known outer membrane perturbing compound like polymyxin. And so enter Craig McNair, who was very interested in trying to test this in an animal model of infection. This is a checkerboard analysis, of course, of the synergy between gram-positive antibiotic novobiocin and pentamidine. You'll note pentamidine doesn't have growth is dark here, and growth inhibition is white. So pentamidine itself doesn't even have much of an MIC in these conditions, but is very shows this characteristic staircase of synergy against A. baumannii, particularly with this molecule novobiocin. And so Craig's work in the animals showed that together these two compounds led to 100% survival of all individuals in this mouse model of infection. And if we look closely at one of the organs, he looked in all organs, and all look pretty much like this. Spleen shows, of course, high numbers of counts in the PBS control, the novobiocin dose, and most of the individuals receiving pentamidine. And the combination, we saw complete sterilization. This was after allowing outgrowth for a couple of hours before treating with any of these treatments. But we see, of course, a spectacular effect of this combination. And these concentrations, by the way, 10 mg per kg and 5 mg per kg, are dose sparing in terms of what is typically given for both pentamidine and novobiocin. So this was pretty exciting stuff to us. Again, this came from just a pilot screen on a small number of compounds, and we've now expanded the screening effort. This is being led by research associate in my group, Mayafara, where we're screening, frankly, our entire chemical file, along with natural product extracts, looking for more compounds with this phenotype. We're looking for compounds. We believe in 3 different mechanisms. Those, again, that would be membrane active, like pentamidine, outer membrane selective compounds. Again, pentamidine is kind of in that category, and but we're also got our heads up for outer membrane biosynthetic inhibition compounds that would be among those from the screen. I want to move now to our work in chemical interactions, specifically nutrient biosynthetic metabolites. Here we're really interested in the genes important for E. coli survival. Maury, I mentioned earlier, showed that some 303 genes were essential for growth on rich microbiological media. And it turns out that if you ask this question on minimal media, you come up with a larger set of essential genes, 119 additional genes that are important for growth on nutrient-limited media. And of course, the genes that are important here are dominantly in 3 areas: amino acid biosynthesis, nucleobase synthesis, and vitamin and cofactor synthesis. And our interest in this area, of course, is thinking about whether or not these particular classes of targets could be useful in antibiotic drug discovery. So a proof of principle screen was done in this area a few years ago in my group by Sumaya Zlitny. She looked with a relatively small compound collection of about 30,000 compounds for growth inhibition on just glucose and ammonium chloride. 340 were active under those conditions. And when she added back vitamins, nucleobases, and amino acids as a pool, she found that only 71 of them were active. So she threw away the ones that were active in rich media and prioritized those that were active only in nutrient-limited conditions, and came up with a very creative chemical-chemical interaction approach to generate hypotheses for the action of those compounds. And that was what she dubbed the metabolite suppression array, where we have growth inhibition in M9 with glucose and ammonium chloride, and we can now add back specific amino acids, nucleobases, and vitamins, and then pools and other combinations thereof to generate hypotheses for which metabolites were suppressing the action and where the targets might be. This was a very successful approach, and it came up in this small screen of 30,000 compounds with actually 2 novel compounds. This molecule 379 that was suppressed by pyramidinobenzoic acid and targets folic acid synthesis. And this other compound 13772 was suppressed by biotin and turned out to inhibit BioA of biotin synthesis. We've done a fair bit of work on this, including developing an animal model for the specific for biotin synthesis, and showed and are kind of underway with some more extensive SAR and that involves also studies along with co-structures with BioA. But suffice it to say, we quite like this approach and are interested in finding more such chemicals. And in that context, I'll tell you a little bit about some gene-gene interaction work that we've done that has gone some distance to help us consider new approaches to finding such compounds. So again, here we're looking at the Kale collection of some 4,000 gene deletions. This work was led by J.P. Cote with the help of Sean French. J.P. is now a professor at Sherbrooke University in Canada. And J.P. was interested in trying to look at the network of nutrient biosynthetic genes in the context of the genome. So the idea here was to make a genetic cross of some 82 of 100 or more genes that I noted earlier, which are important for growth on nutrient-limited media, and to make double deletions with each of the remaining 4,000 or so genes in the KO collection. Pin those then on rich microbiological media, selecting for the 2 drug markers, and we would get a sense under rich conditions whether or not there were any synthetic lethal or synthetic sick interactions that came out of that. And remarkably, these gene-gene interactions were just chock full of synthetic sick and phenotypes, and shown here again is an interaction of those 82 genes with the rest of the E. coli genome. There were some 160,000 double deletions made, and their growth recorded. And we get about 2,000 interactions for these 82 genes, on average about 23 interactions per gene. And so it's again, a highly dense network, lots of unknown function genes in here, but again, a remarkable look, I think, at the density of a network that is involved in supporting the growth of these. Again, recall that we're targeting in genes that are important for nutrient synthesis here, but we're looking at the growth on media where these otherwise don't have a phenotype, except when deleted doubly with other genes here. And so one thing that we did notice here was that in the case of lethal interactions, these really stuck out like a sore thumb. We're talking about deletions in a transporter as well as biosynthesis. So on rich media, you can either synthesize a vitamin nucleobase or amino acid, or you can transport it. But if you lack the transporter, then suddenly biosynthesis becomes indispensable. And this was very interesting to us. You know, we noted that the biotin transporter was one of our key synthetic lethals here. And Garima Kumar came up with the following idea. It was a postdoc in the laboratory at the time. We've been struggling with trying to use our metabolite suppression approaches to understanding the action of natural products, natural product extracts. Those extracts commonly contained things like vitamins, nucleobases, amino acids, which had confounding effects on our suppression studies. And so here's a look, however, at the ability of that synthetic compound that I referred to earlier, MAC 13772, to inhibit the growth of E. coli in the context of a biotin transporter deletion. Whereas wild type, of course, was completely unaffected by this biotin synthesis inhibitor on rich microbiological media. These two phenotypes gave Garima a very targeted screen for anti-biotin compounds in the extracts of natural products. And so we got 1,000 extracts from our good neighbor Jerry Wright and looked for growth inhibition in the context of the bio-P transporter. Some 22 extracts lit up in that assay, of which 10 of these were active against wild type E. coli, and that allowed us to prioritize 12 extracts that we believed contained anti-biotin compounds. And so large scale culture, extract, and purification led to the rediscovery of these two compounds here. These are meclanomycins, they're called, were discovered some years ago in a Japanese lab. They have this unusual cyclopentadiene activated amine here. And for the chemists in the audience, you may have noticed that our other antibiotic compound was also a kind of a soft nucleophilic character. It had a hydrazine moiety, and it turns out these compounds work in exactly the same way. Two different dipeptides here, of course, valine and isoleucine and methyl derivatives of these, and these are also really interesting inhibitors of BioA in biotin synthesis that we we continue to work with today. So we think these targets are quite interesting, targets in nutrient stress, and we think that they mitigate innovation risk. So that this is really a look at kind of a Venn diagram of what it takes to support life In nutrient-rich conditions, we have this set one, and folks like me, academic drug discoverers, are talking a lot about the set that is set two, a much larger group of targets that are important for growth in say an animal. And those, working in biotech and pharma, remind people like me that, the innovation risk of working with that in vivo set is very, very high compared to the nutrient-rich set. And that, of course, is because we have things like in vitro assays. And clinical microbiology labs work through, understanding growth inhibition on media. And so it's occurred to us that, this collection of nutrient stress targets are probably somewhere in the middle, because we have good in vitro assays, nutrient-limited media, for example, to try to understand the action of these compounds. But of course, in some cases, those targets will probably lie outside those targets which are necessary for growth in vivo. But we're interested, of course, in these areas where we have viable targets and they have low innovation risk. Okay, I'll finish off today with a kind of a brief look at something that we're doing a lot of more recently, looking at the chemical, genetic or chemical promoter interactions in this case, using this collection out of Yuri Alone's laboratory again, which is fusions of some 2,000 or so promoter regions of E. coli with fast folding green fluorescent protein. Of course, using our panning robotics, we're able to generate a beautiful fluorescent array on solid media. And we had one problem after we did that there was no instrument to image this array. It wasn't lost on us that you have a very signature response of the promoters of E. coli that is illuminated through all of these green fluorescent proteins, and that we'd be able to put different compounds in this media and try to chart chemical genetic interactions on a genome scale. But unfortunately, we had no way to image an array such as this. So once again, Sean French, who's our master inventor in the laboratory, set about to create this particular tool. We call it the printed fluorescence imaging box, or PFI box. It is a 3D printed box that keeps the light out, with a Raspberry Pi computer that records data from a CMOS type or iPhone type camera that is in this box, that is targeted towards an array of colonies at the bottom of the box. Here's a relatively low density array. It is sitting atop of an LED array to excite the fluorescence in GFP here. And of course, this is a pretty straightforward thing to print these kind of boxes. It's I dare say a real bootstrap approach to creating a tool like this. And in fact, these drawings and code are now open source. Any kid can print this in her garage and make a PFI box. And here's a look at Sean assembling one of these. So the first thing to go into the box, of course, is the array of LEDs. The box is assembled pretty simply, and you can see at the top here is where the iPhone type camera goes, along with the Raspberry Pi computer. These boxes we can print for about $200 a piece, and so we're printing lots of them. The Brown Lab now has no less than five printers to print these in 3D, and we're collecting data now with really in a parallelization mode. And so, as I mentioned earlier, our goal here is really to get a sense of the signature expression in response to to chemical compounds like antibiotics. This is a linear discriminant analysis. So what we've done is we've keyed on the 3 LDA parameters that describe most of the variants. Here we're looking at more than 100 different antibiotics in various having various mechanistic classes: cell wall, nutrient stress, membrane active compounds, those at the folate pathway, protein translation, DNA replication. And probably what's not lost on this crowd is that we see quite a cluster and signature responses that are characteristic of the mechanistic classes of the compounds that we're analyzing using this PFI box. And so we're doing a fair bit of this these days, building out a very large training set of known antibiotics, and interested in comparing that the responses of that training set to novel compounds that we generate in our work. And we are more recently using a fair bit of AI machine learning to do this analysis. And out of this is coming some really exciting work on mechanistic prediction of for example, these novel compounds that we put into this deep learning model. I'll end with a quote that I think is just fantastic, and I don't mean to be dismissive of systems research, as I call myself one now, but Roberto said not long ago, if I couldn't be a scientist, I would have been a systems biologist. And but clearly this has been an area that has really kind of changed the course of the work in my laboratory, and I'm pretty excited about our efforts in this area right now, and happy to be called a systems biologist. I work with just a fantastic group of young people. Of course, this is the way that we assemble these days, which breaks my heart. I have a whole bunch of funders. These are folks that have funded me, and in some cases I have consulted for. So this is both acknowledgement and disclosure. I think I mentioned many of the trainees that were involved in this work, but there's a long list here. And I should highlight my collaborators, Brian Coombs and McMaster, who's kind of taught us everything we know about how to do animal models of infection. Deb Hung at the Broad, who's helped us with our work in the pentamidine area. Here at Tatamori, I mentioned throughout the talk. Whitfield at Guelph has really been super helpful in the area of outer membrane synthesis, LPS synthesis in particular. And Jerry Wright at McMaster, who's been my longtime collaborator in the area of natural product extraction, isolation, purification, et cetera. And thank you. It's a pleasure to be here. I'm happy to take any questions.
Wonderful, Dr. Brown. Round of applause. Before I answer questions from the general audience, I thought I would give a chance for the panelists to ask questions first. So Katharina or Helen, if you have a question.
Yeah, it was a wonderful talk. I appreciate very much also the engineering aspects of the research that you do. So I was wondering, that's curiosity for application. So are you able to commercialize some of those hand-built things that you have in your lab?
Yeah, it's just that's a fun question, Katharina. We've been thinking a fair bit about the utility of, for example, this imaging box, and I mean, what we sort of just elected to do instead is just make it available to anyone by making all that open source. I think John alluded earlier to the difficulties in this research area around commercialization in general. But I guess I'm hopeful for the future that devices like this and approaches like this will be important ultimately to the discovery of new antibiotics. We're living in a time where there's a great deal of hand-wringing and interest in how to how to make antibiotic drug discovery viable. And, I'm really encouraged by, some of the advances we're seeing, in Europe, and discussions in the United States in particular, big markets that could make antibiotic drug discovery a bit, quite a bit more interesting to investors. But in the meantime, we're going to keep doing it, and and try to make some inroads with developing leads and new avenues.
And Helen, if you had a question.
Yes, a great talk. So the question is about GFP fusions, promoters. So did you compare your data with RNA-seq, for example, exposure to different classes of antibiotics? Do you see significant overlaps or completely different? What's going on?
Yeah, we do see some similarity, Helen, for sure. Something that we really like about, so we do a fair bit of this. Maybe I'll take a step back in terms of just trying to come up with, systems approaches to trying to generate hypotheses for mechanism. Done a fair bit with, chemical genetic interactions with the deletion collections. You can do overexpression, which is something I didn't talk too much about. Those two things work some of the time, actually infrequently, I think, relative to, all the compounds that we can throw at them. But this fluorescent array has really turned out to be very, very robust in terms of predicting mechanism of action. And I think it's in part because we're able to collect kinetic information. So we can image that array over time, right, over the duration of growth of the array. And so it's I think it's a real advantage over RNA-seq, where it can be done. Of course, you can collect a lot of time points by RNA-seq, but it's it's relatively clumsy by comparison to, and expensive to generate data that way. So I think that's probably the advantage. But to answer your question, I think in general, sure, we see lots of similarity between the responses that we get from our array and those that you would get from any say given time point with an RNA-seq experiment.
We have a question from Professor Kim Lewis. Eric, have you considered interrogating essential versus essential gene interactions?
Yeah, so there are. We're doing, Kim, we're working mostly these days in trying to do essential gene versus non-essential gene interactions. And as you'd know, that's also pretty tricky, right? It involves usually coming up what's necessary. And of course, the linchpin genetic constructs that have to be made there are conditional mutants. There's been some really great work in the yeast area, done principally by Charlie Boone, Brenda Andrews, in making temperature-sensitive mutants to do that kind of work. They showed, for example, there was a really, a much even higher rate of genetic interactions among essential genes. That CRISPR has been the approach that we've been taking to this. We started out doing this kind of work with CRISPR style approaches, and I think that's going to be the way to do it. Ultimately, yeah, essential gene is a really exciting area to think about, particularly where you might have some predictions about how common antibiotics and maybe not so common antibiotics might be used together.
We have a question from Jitender Mela. Professor Brown, as we know, several genes or proteins and their interactions are conserved across different bacterial species. Did your group test any final small size test set across different species? I mean, can we expect the gene-gene or gene chemical interactions are also conserved for conserved genes and proteins?
Yeah, that's a really interesting question. And of course, to do that work across species is a work in progress for us. There are a lot of gene sets which are now available, largely, ordered transposon mutation collections that we can do that kind of work. And I won't say that we've done a comprehensive analysis, but we have done some work and I can tell you that, we see different interactions from different organisms. One area that we've done, and it's a paper that we published some time ago, actually, it was a collaboration with Jerry Wright some years ago, where we went looking for combinations of chemicals, tetracycline and other drugs, and their action on Pseudomonas E. coli and Staph aureus. And we saw unique chemical interactions, for each of those organisms. And it makes me wonder, frankly, whether or not that, plus the work that we've been doing more recently, that you've got a great deal of conservation among, particularly among housekeeping genes and bacteria, and to a lesser extent, a fair bit of conservation around typical biosynthetic pathways, et cetera. But the way the organisms are wired, right? These various pathogens may turn out in the end to be something which is, perhaps quite specific to a pathogen.
We have a few more questions, and some we might not be able to get to. Nora Vasquez Laslop asks, thanks for the great talk. First of all, makes a comment. What's the advantage of monitoring colony growth instead of growth in liquid culture in the high density arrays?
Yeah, no, that's a great question. So we do this stuff in liquid as well as in solid. It just turns out that it's just so much easier in order to pin at high throughput. We can reliably, of course, prepare a 384 well plate in liquid media. It gets a fair bit more challenging to do that in 1536. And as you saw, of course, we're able to pin, on a microwell, uniwell size plate at extremely high density, greater than 6,000 clones at a time. And so it really just makes our life a whole lot easier than working in liquid.
We have another big thank you, Dr. Brown. This is from Andrew Hogan. It was particularly interesting to see the DIY approach to fluorescent imaging. As for the interaction screening, most of your work and others focuses on synthetic, lethal, sick interactions. On the other hand, is it that synthetic viable fit interactions are less common or less informative? I was wondering if you could give your thoughts on synthetic viable interactions.
Yeah, I love this question. We're actually really interested in synthetic viable. You may have noticed our vancomycin antagonism screen, right, is based on a synthetic suppression mode. We really like that for chemical screening. So to identify, and also work that we've done, other work we've done in my lab around maltoic acid synthesis, we've taken advantage of synthetic viability there to look for compounds that are active on that pathway. So we love this phenotype. We have done some work. It involves, as Kim alluded to earlier, in creating conditional mutants, right, in essential genes and looking for the suppression of their essentiality. It's quite a bit more demanding technically, but a very exciting area. And I think I do think those interactions are more rare, however.
Thank you so much, Dr. Brown. What a wonderful talk. Thank you so much. Round of applause from Internet land. Dr. Brown, you had one more question from Ohad Mir. If you could go ahead and answer that for him, I would greatly appreciate it. I'd like to introduce Professor Helen Zgurskaya. She's the George Lynn Cross Professor of Chemistry and Biochemistry at the University of Oklahoma, go Oklahoma, where she leads a research program on antibiotic discovery and resistance, multi-drug efflux mechanisms, and efflux pump inhibitors. I especially appreciate that aspect of her work. She is the founder of the university's Center for Antibiotic Resistance and Discovery, an international team composed of 13 research groups spread across 5 academic campuses in the Oak Ridge and Los Alamos National Laboratories. Professor Zgurskaya has written more than 80 papers, several books, and is a co-editor of a book. She is also an associate editor of the ACS Infectious Disease Journal and a member of the editorial board of Antimicrobial Agents and Chemotherapy and Frontiers in Microbiology journals. The title of her talk is going to be The Two-Faced Janus of Multi-Drug Efflux Substrates and Inhibitors. And with that, Helen, take it away.
Thank you, John, for introduction. So maybe just first Janus itself. Janus is the god of beginnings, transitions, time, and duality. And Jupiter himself can move, force it back because of Janus's work. So I'm going to try to convince you that Janus is the god of bacterial cell envelopes. So beginnings, right? So if you think about permeation, first what comes into mind is that bacterial cell envelopes are highly diverse. They're diverse chemically, structurally, and so on. However, the principles that define permeation of small molecules into the cell and back are actually the same for diverse envelopes. So in my lab, we're trying to understand what are these principles and rules. So transitions, probably one of the most interesting aspects of what's going on with bacterial cell membranes. And perhaps the easiest illustration is to look at gram-negative cell envelope, where you have two membranes, the outer membrane and the inner membrane, and efflux pump transporters that actually function in the context of these two membranes. So if there is no efflux and you add a drug into a medium, what you expect to see is the linear dependence, linear increase of internal concentration of the drug. However, if efflux pump is present, then it will change the shape of this curve. At low drug concentration outside, pump is effective, so it will reduce internal concentration of the drug. However, if you keep increasing the concentration outside, eventually you can overflood the pump and the drug will rush in, and eventually you will have again linear dependence. So however, this scenario, okay, when you can flood the pump and get drug inside the cell, is possible only if the influx of drug molecules across the outer membrane is fast enough. So the influx must be faster and more efficient than efflux. However, if opposite is true and the drug molecule gets across the outer membrane is very slow, then efflux is very powerful. In this condition, you have this transition, so-called bifurcation, where you have completely different regime of drug accumulation inside the cell. What happens in this case, you keep increasing concentration of the drug outside. However, you cannot saturate the pump and you cannot break the barrier. So these transitions in drug permeation are governed by efflux constant, which is thermodynamic measure of efficiency of the efflux pump, and the barrier constant, which is the ratio between maximum influx and efflux. So once these transitions are in play, right? So the system is actually a little bit more complicated. So in addition of two membranes and single efflux pump, what often seen, not often, normally function inside the cell is additional transport system, permeation across the outer membrane, uptake transporter, efflux transporters, and so on. So we spend a lot of time trying to understand how all these systems work together. So if you have two transporters acting across the same membrane, like for example, this tripartite efflux system pump, so you will see additive effects on MICs or drug accumulation. However, if transporter function across two different membranes, like MDFA and ACRA beta or C in E. coli, then you can expect to see synergistic effects. However, it's not all. So in addition to this interaction between different transporters that actually controls the passages of molecules, right? You also have interactions with the permeability barriers that I showed you on the previous slide, right? So in this case, transporters are synergistic with transmembrane diffusion. However, it only if the fluxes occur across the same membrane. So this transporter will be synergistic with the outer membrane barrier, and this transporter with the inner membrane barrier. So often people ask why these inner membrane transporters are even important. They are important because if drug molecule cannot penetrate across the inner membrane or penetrate slowly, these transporters become very efficient. So the same applies to enzymes. Enzymes in mathematical terms, this depletion of drugs by enzymes is no different than efflux. And of course, in this case, synergistic and additive effects act with the barriers and efflux pump as well. So, and finally, we also looked at some influx, and influx, when it is more efficient than efflux, compounds will accumulate inside the cell, but it's only under this condition. So efflux pumps, which are the most effective, are those from resistance modulation, superfamily of transporters, because they're able to pump drugs across the outer membrane in gram-negative bacteria. More than 20 years were spent trying to understand the structure and molecular mechanism of these transporters. These are trimers, right? And each protamer has the structure indicated here. There is transmembrane domain, pore domain, funnel domain. There are at least 3 drug binding sites. Transmembrane binding site between transmembrane segment 1 and 2. Access pocket, that's where the drug enters the pump, and then deep pocket. So to pump drugs across the outer membrane, transporter associates with membrane fusion proteins, shown here. So they're very structurally dynamic proteins, conformationally flexible. So MFP and R&D pumps have stable complexes in the inner membrane, and in this state, their membrane fusion protein is highly flexible and moves. And only when the outer membrane channel is present, then it serves like a scaffold for membrane fusion protein to oligomerize and form this funnel that enables efflux of drugs all the way across the periplasm and into external medium. So one of the features of R&D pumps, which was intriguing for a long time and also still remains very impressive, is their extreme polyspecificity. So this polyspecificity actually comes from different mechanisms. One of the major reasons why these pumps are polyspecific is the synergy with the membrane permeability barriers and modifying enzymes, those that I just described on the previous slide. In addition, there are also features in the structure of the transporter itself. So if you look at these transporters, I already mentioned at least 3 binding sites. This is the deep binding pockets, and there is access binding pocket here. And there are multiple channels that lead drugs into the binding pocket. Again, at least 3 channels are shown here. So these low affinity substrate entry and binding sites contribute to polyspecificity. And in addition, all these tunnels are filled with water, and water acts as a grease, so it shields unwanted interactions and promote translocation by these pumps. So the polyspecificity, right? One of the major impact of this polyspecificity is impact on MDR in clinics, right? And Acinetobacter baumannii is one of the famous bugs here, and extremely multi-drug resistant. So to figure out how R&D pumps and polyspecificity plus synergistic interactions, right, contribute to MDR phenotypes in clinics, we looked at 3 different strains of Acinetobacter. So they also represent a timeline. Acetobacter was not as visible 20 years ago, but now it's one of the major concerns, right? The susceptible strain that we have is from ATCC 17978, isolated in 1961. And we also work with two MDR strains. One is from Europe, AYE from 2003, and 5075 from US, isolated in between 2004 and 10. So susceptible 17978 strain or produces one R&D pump, ADIJK. And if we now compare the expression of two other IJK and another pump, ADABC in MDR strains, what we will see is that AYE or produces both pumps, and 5075 also or produces both pumps. So the next panel here are actually MIC comparison between MDR strain and susceptible strain here. So what you will see here is that some antibiotics such as azithromycin, ciprofloxacin, gentamicin, and so on are essentially have extremely high MICs in multi-drug resistant isolate. However, for example, erythromycin, SDS, novabiocin do not have increased MICs. And in fact, MDR strains are more susceptible to novabiocin than ATCC strain. We also looked at permeation in the context of the whole membrane. So this is fluorescent probe and PN, and cell envelope of 17978 is the most permeable for this probe, followed by AYE, and the lowest permeability is for 5075. So to figure out what's going on, we knocked out each of the pumps and combination of these pumps in all three strains, and again compare multi-drug resistant isolates and susceptible strain. So again, in this case, we're comparing mutants to mutants. So if we knock out ADIJK in MDR and 17978, we don't see much change in MICs. So MDR strain remains highly resistant to most of the antibiotics. With a few exceptions, we see a little bit change in gentamicin, and that's pretty much it. When we delete ADB pump and again compare ADB mutants, so what we'll see, dramatic drop in MICs. So both MDR strains become more susceptible, with the exception of ciprofloxacin, chloramphenicol. In addition, we see significant increase in susceptibility to zeaxanthin. So MDR strains become more susceptible to zeaxanthin than 17978. Finally, when we look at double mutants, we see again dramatic drop. Ciprofloxacin resistance remains, chloramphenicol resistance remains, because actually these antibiotics are resistant to this antibiotic provided by different mechanisms, target modification and enzyme expression. However, for other antibiotics, efflux is the primary mechanism of resistance in these MDR strains. And in fact, if you knock out these two pumps, MDR strains become even more susceptible than 17978. So because of this, dramatic synergistic effect of knockout of two pumps, recall that I said that if the pumps are pumping antibiotics across the same membrane, you expect to have additive effect. However, we see synergistic, right? Something else is going on. So to figure out what's exactly going on, we looked at structure of the outer membrane, specifically at lipid A. And we didn't find much changes, no modification in comparison to ADB knockout, double knockout mutants. We do see loss of one of the ethyl chain in delta ADEIJK. And from metabolics and other studies from other labs, we know that ADEIJK is involved in lipid metabolism. So we did RNA-seq analysis, and what we found is that ADABC actually and IJK knockouts have completely non-overlapping transcriptional response. They're equally distant from their parental strain, and ADAB significantly dominates the phenotype of double knockout mutants. We identified several interesting efflux pumps which are involved in suppression. They are overproduced in efflux knockout strains, and we see very peculiar changes in transcriptional regulators, which are specific to ADAB, IJK, both pumps, and so on. So these studies also led us to try to identify what are the substrate specificities of AD, ADC, IJK, and the third pump, which is present in Acinetobacter. This pump is FGH. So in our studies, we're using the set of 4 strains. What is the reason? So you can change the efflux by inactivating one of the components, deleting the entire pump, right? What happens if the barrier remains, if the outer membrane remains? You cannot really judge what is more important, permeation across the outer membrane or efflux. So to increase the influx of the drugs, we break the barrier, we put the pore, increase the efflux, and this allows us to look at, when we saturate the pump with the drug, which substrates are the best. So, and this allows us to assign substrate specificity. IJK is the most effective against beta-lactams, coumarins, and cyclins. ABC, aminoglycosides, macrolides, and zeaxanthin, and FGH, trimethoprim, and phenycols. We use similar approach with Pseudomonas aeruginosa. So Pseudomonas is a little bit more complicated than Acinetobacter because it has 12 R&D pumps. I have here 7. We were able to assign to many of these pumps substrate specificities. They're similar. And we also know that, for example, in laboratory conditions, MXAB or PRM is the major pump which is expressed. We also know that tri-ABC pump, which is specific to triclosan, is expressed under laboratory conditions, as well as McGHI or PMD pump, which is growth phase dependent and involved in secretion of phycyanin. So Pseudomonas is challenging bug. And of course, if you think about efflux, right, one of the major questions is, okay, so we know everything about efflux, what can we do about it? So how we can inhibit efflux pumps? So the mechanism is complex, but can be boiled down into 3 basic steps, right? So you have complex between R&D and MFP, which is ready to grab the drugs, and then assembled when substrate is bound to the transporter, and finally open or extrusion conformation when the drug can be pumped out across the outer membrane. So theoretically, what you can do, you can interfere with pump assembly, prevent complex formation. You can inhibit R&D transporter by competitive inhibition, conformational locking. You can also interfere with proton translocation. And finally, you can block the channel or use antibodies or phage on the outside the cell, try to interfere with the pump. So I will tell you a little bit about these 2 mechanisms of inhibition. So we teamed up with Los Alamos group to try to identify efflux inhibitors that will prevent complex assembly. And we identified several structurally unrelated compounds which are able to do that. They bind to ACRA, E. coli protein, membrane fusion protein, and they essentially interfere with the complex assembly. How do we know this? So because the complex is in the periplasm, you can put trypsin into periplasm and digest ACRA. So, and when the complex is fully assembled, you see characteristic profile. If there is no TOL-C, then the profile changes. And we can see that in the presence of some of these inhibitors, we see the change in their profile consistent with the defect in assembly. So we also identified their binding site, and for this purpose we used tryptophan fluorescence, scanning mutagenesis first and then fluorescence. So ACRA has a single tryptophan that we removed and replaced by phenylalanine, and then we introduced phenyl tryptophans along the protein structure, and we were able to identify the binding site for the inhibitor. It is between these two domains, which are responsible for assembly of ACRA into this funnel. So we think that maybe this inhibitor indeed prevents assembly of the complex. There's another group of inhibitors that we're interested in are inhibitors that act on the transporter itself. Again, different groups, this time isolated, structurally unrelated molecules that act as inhibitors of the pump. So the first was discovered by microsites, specifically Olga Lomovskaya's group, and now broadly used in different studies as an inhibitor, broad spectrum inhibitor of efflux pumps. Then MBX 2319 followed, which is enterobacterial efflux inhibitor, and act again E. coli and several other enterobacterial transporters. So what happens to these inhibitors? Everyone believes that they bind in so-called hydrophobic trap, which is located in so-called deep pocket. So recall I told you that there are at least three binding sites, but in periplasmic domain there are two. Access pocket is the site where drugs and inhibitors enter the pump, and then they reach and bind in the deep pocket. So the hydrophobic trap is a set of phenylalanine rings which trap through the stacking interaction, trap molecules, and if inhibitor bound there, pump cannot rotate out of the conformational state, and the efflux process is inhibited. So here we see again the influence of genus, because what happens if you look at crystal structures of ligand-bound transporters, what you will find is that inhibitors, in this case, this is MBX inhibitor, bound in the hydrophobic trap. Take a look, stacking interactions of phenylalanine rings that keep inhibitor in the binding pocket, and now different substrates. And what you'll see is that APIs and substrate have overlapping binding sites. And most inhibitors, including inhibitors that we discussed that target ACRA, are actually also substrates of efflux pumps. So the question that bothers us and everyone, so what is the difference between efflux avoiders, substrates, and inhibitors, and how we can make better inhibitors? So the study was done using 260 Rempex compounds. These are derivatives of the first inhibitor, broad spectrum inhibitors identified by microsites, and the series were developed by Rempex and currently Apex by Pharma. So what we did is shown here. So these are peptidomimetic antibiotics. Essentially, they do have antibacterial activities. So the first is to measure MIC or IC50 in 4 different strains, so that we can account for the efflux and permeation of the and the permeation of the outer membrane in activities of these antibiotics. In addition, they are inhibitors. So inhibitors of efflux, you can take a look at by measuring minimal potentiating concentration. Levofloxacin, norobiocin were used, and this is typical checker border assays that Eric already told about. You can also measure efflux inhibition in the bacterial growth independent way. In this case, you can look at the accumulation of fluorescent probes. you will see HELX 33342. And in this case, you're looking how increasing the concentration of inhibitor in their external buffer increases the accumulation probe inside the cell due to the inhibition of efflux. You can also look at physical interaction, direct physical interaction with MXB, the major efflux pump of Pseudomonas aeruginosa. So once you have antibacterial activities, what we did, we essentially looked how they correlate between, if there is a correlation between growth inhibition due to antibacterial activity and potentiation. So compounds have wide variety of MICs. Some are affected by both efflux and outer membrane. So when you see a significant drop in double, knockout, and or expressing strain, you see significant synergy. Or some of them are efflux affected only and others are not affected at all. And there is not much correlation between permeation, effect of permeability barrier, and antibacterial activity of these compounds. So what you're looking at here is IC50 is the most permeable strain and the ratio between the two. And so there is a trend, but no real correlation. Also, there is only weak trending between potentiation of levofloxacin versus antibacterial activity of these drugs. And this result suggests that essentially antibacterial activity and efflux inhibitory activity are are actually associated with two different features in these compounds. So compounds vary significantly in inhibition of fluorescent probe efflux. So this is again bacterial growth independent assay for inhibition. We analyze this, and if you compare now in efflux inhibition and growth dependent and independent assays, again, very big correlation between the two. However, there is negative trend, which is consistent. So at least some relationship between the two activities are there. So what we did with this first clustering of the activities, and what you can see here is that avoiders, compounds that are not recognized by efflux pumps, efflux pump inhibitors, substrates, and compounds that combine the properties of two group separately. They occupy different spaces. However, the space is not really clearly defined, and they meet in the same quadrant of principal component blocks shown here. So once you have this, the next question is, what are the physicochemical properties that associated with efflux inhibition, antibacterial activities, and so on? So we looked at the structures of molecules, and they have this characteristic polarizable features. So they have hydrophilic end and hydrophobic end. They have also dipole moment, and these features are also important when you will see a little bit later. And of course, the shape of the molecules is also contributing a lot to permeation. If you look at some of the properties of this inhibitor, they're completely different from antibiotics. They cluster separately, and they do not really overlap with antibiotics. So they permeate rapidly the outer membrane. The efflux of the contribution of outer membrane is very low. So this is the model of the outer membrane. This is outer leaflet with LPS and inner leaflet with phospholipids, and molecular dynamic simulation of one of the Rampix molecules moving across the membrane. So interestingly, these compounds differ from antibiotics in how they interact with the outer membrane. So these dips in potential of mean force graph indicate where the energy barriers are in this membrane. And as you can see, antibiotics have two barriers in core and the hydrophobic part of the membrane. However, Rampix compounds transfers the membrane, their energy profile is flat, so there is no significant barrier when the molecule moves across the hydrophobic part of their membrane. And if you look at molecules, they're actually quite hydrophilic. So it's interesting how they do this. And simulation suggests that they actually flip, more like lipids flipping across the membrane. And they also bring a lot of water with them when they do this. So how you can describe interactions of these molecules with membranes, right? I think that these are important descriptors that we need to account for. Outer membrane was split into 7 different slabs, and these interactions of Rempex compounds with different slabs were enumerated through the hydrogen bonding, membrane diffusion, and several other descriptors. So the next, once the drug Rempex compound passes the outer membrane, it is recognized by efflux pumps. So this also, we try to account for these interactions on their experimental level. Surface plasmon resonance was used to monitor the interactions, and compounds again have broad variability in affinity to the MXB. On the molecular level, again, we're coming back to the transporter, access pocket, deep pocket. So the Rempex first must bind to access pocket and then, driven by affinity, enter the deep pocket. So there are multiple interactions in both sides of transporter with these drugs, right? So, and these interactions in drug binding pockets are specific to the structure of the molecule. So we describe molecular interactions with MXB by counting the contacts within these 2 different pockets and calculating overall affinities to access and deep pocket of MXB. So when you have all these different descriptors, right? So what happens often, you have the same property described by multiple descriptors. How you get rid of this? Because some of the properties can be overweighted in models, because you have, for example, 30% of descriptor describing the shape, but you already have the shape there. So we did clustering analysis. So descriptors that I described on previous slides were about 170. They went through clustering and selection. Total 29 clusters were identified, and then out of those 29 clusters, a single descriptor went into machine learning to generate models. So the 2 models I will show you here, I have to mention that, recall that these compounds do not have problems to permeate the outer membrane. So we could not build the model for permeation across the outer membrane, but we could account for the interaction with membranes by looking at the efflux avoidance inhibitors. So what you see here in green are descriptors of interaction with the pump. And you can see the efflux inhibitors and substrate actually have positive correlation with certain interactions in access pocket and specific residues of MXB. If you look at efflux avoiders, interaction with pumps are different, and you have both permeation descriptors as well as physicochemical properties that define the properties of both efflux avoiders and inhibitors. So to see if the models actually are functional, we calculated these descriptors for a library of about 670 compounds. These are unknown compounds and also antibiotics and traditional efflux inhibitors and traditional antibiotics. And we identified avoiders and inhibitors predicted based on the properties that I just described. So avoiders are mostly antibiotics from cephalosporin, lactams classes. And indeed, we checked efflux ratio is very low for these drugs. Inhibitors, surprisingly, and we were very pleased to see the top inhibitors that were identified were actually from Basilea. These are efflux pump inhibitors, which were patented and recently described by this company. In addition, fluoroquinolones were part of this group. So fluoroquinolones actually are drugs that interact with hydrophobic trap in transporters, and they do potentiate beta-lactams in gram-negative bacteria. So in this case, hopefully, maybe we think that maybe this substrate is also inhibitor. So, so when you know this, what else you can do? So we went back to inhibitors that we identified. I show you two of them. So SLU 258, we did some med-chem follow-up on these compounds and were able to a little bit improve their activities in E. coli. However, if you look into Pseudomonas, they are not effective in potentiation of antibiotics in Pseudomonas cells. And if you look at descriptors that were identified, cylindricity, interaction with specific amino acid residues in MXB, aromatic ring content, hydrogen acceptor, log D, and polarizability, actually they are far away from efflux inhibitors that we know about, right? So, however, if you modify them by addition of dihydroamidazoline, being here, so you bring the properties of molecules closer to those that we found for Rempex compounds. And we do see potentiation now of antibiotics, carbamazepine, levofloxacin, and so on, in Pseudomonas aeruginosa. So the same for the second analog 17O. Again, their original molecules that worked in E. coli is far away from Rempex compounds. We brought them closer. So green shows that we move this descriptor closer to the optimal space, and red when the descriptor goes away. So we moved closer again to Rempex compounds, and again we got potentiation of antibiotics in Pseudomonas, and we demonstrated this also for bromophenolphthalein substituted analogs, which are very hydrophobic and favorable in terms of these properties, but could bring them closer and see potentiation again. So to summarize, what I described and told you about is that diffusion barriers and enzymatic inactivation in combination with efflux pump can generate additive or synergistic changes in intracellular concentration of antibiotics and, of course, their antibacterial activities. So this synergistic effect of combinations with enzymes, especially for the barrier, permeation barrier, is underappreciated, and hopefully this will come into light now. So polyspecificity is product of synergistic interactions with outer membrane mechanisms, outer membrane and other mechanisms of resistance, and of course, plus structural features of transporters. However, despite this polyspecificity, R&D pumps have specific substrates, and those can be identified. Why it is important, right? So if you want to inhibit the pump, you need to know which substrate is actually the best, right? Because then you can look at the interaction of the substrate with the pump and go for the best inhibitor. So structurally diverse compounds possess efflux inhibitor and substrate often are in the same chemical scaffold, right? So multiple studies identified structurally diverse inhibitors, but it comes also from the same polyspecificities that you see for substrates, because they go into the binding site and inhibition happens there. Efflux ligands, they have specific chemical properties, and they are distinct from efflux avoiders, and I hope that this will help in the future with optimization. Models are predictive, and they select different chemical classes of compounds, and I hope I convince you that genus is the gut of bacterial cell envelopes. Acknowledgements. Studies that I showed you were generated by present and past members of the lab, Jitendra Mehra is contributed to identification of the rules for efflux avoiders and inhibitors. Inga Leos, Justina Adamyak worked on Acinetobacter profiles. Mohammed Manir Zaman working on efflux inhibitors and E. coli. And collaborators were acknowledged on the slides. This is really an interdisciplinary project, and a lot of communication and collaboration is going on. Olga Lamovskaya from QPEX, Jörg Dreyer from Brasilia for compounds, funding from NIH, and thank you so much for your attention.
Round of applause for Professor Zgurskaya. If any of you have questions, please put it in the question and answer. I'd like to ask the panel if they have questions first.
Yeah, well, okay, Eric, go ahead. I'm sorry.
Go ahead, Katharina.
Okay, thanks. So Helen, that's of course beautiful if one can kind of predict combinations of drugs, inhibitors and efflux pump blockers and so on. And I was wondering if you already looked at the combinations with metal ions that could also help to block or inhibit or whatever. Is there, I think there should be also some combined synergy, but I'm not.
Yes, I would expect this, right? Yeah, I would expect there should be some synergy, but honestly, we never looked at metals. Okay. So yeah, we need to look at them. I agree.
Yeah, thanks. It's beautiful.
Helen, that's fantastic talk. I keep wondering if there's so little natural product discussion around efflux pump inhibition. And yet it just seems just so obvious that nature, you know, should be trying to exploit this. I mean, the redundancy in transporters may be an indication, I guess, of a kind of a tug of war between, you know, the production of such compounds and the defense against some. And I just wondered, and on the other hand, you know, your work with known antibiotics, you know, antibiotics often being substrates. And I just wonder, is it possible that's kind of slipping our attention? That is, is it possible there are weakly active antibiotics, which are actually much more potent efflux pump inhibitors?
Yes, actually, this is one of the questions that bothers me, right? So I think that there should be antibiotics which are ideal substrates, right? And what happens with ideal substrate? It has this high affinity to deep binding pocket. So it has high affinity to access pocket, to deep binding pocket. So it will have this preferable pass through the pump. And if you have a combination of such antibiotic and another one, right, that you want to get and kill, right, you will see efflux inhibition. So the bad substrate will have pass into the cells, the good substrate will be pumped out. So this is something that I think we need to look closer at, what exactly is going on. So I know that in the field, a lot of people believe that binding to hydrophobic trap actually blocks the pump, so it stops. It doesn't rotate out of the this bound state and so on. But I don't think so, actually. I think that because a lot of substrates also interact with hydrophobic trap, it's more complicated than, because most of the efflux inhibitors that we have, they actually potentiate only some of the antibiotics and not all of them. If you block the pump, all must be potentiated. If they are not all that are potentiated, then something else is going on. So I think that we will figure out what's going on eventually, and natural products will play important roles there. I'm pretty sure that those natural products are the best substrate in inhibitors.
Thanks.
You know, Helen, Stuart Levy's been on my mind recently with his recent passing. And I know that Paratech finally has come up with a compound that they're utilizing, a tetracycline derivative for susceptible bacterial infections. But his dream was always developing that tetracycline efflux pump inhibitor, and actually bringing it to clinic. And watching your talk, it's clear to me that you've answered every question I think that I would have had. You're so complete in your presentation. So I wanted to ask you a question. How far are we from an efflux pump inhibitor coming into clinical practice?
Very far.
Well, that's not good to hear.
So there are multiple issues, right? The major probably is that they're toxic. So that's one question, right? With Rempex compounds, because of those primary amino groups, right? Accumulation in kidneys, immediate, and so on. Actually, to give the credit to Qpex and Olga Lamovskaya, right? So they actually bypassed this toxicity, and they bypassed it by making a soft drug, which is one of their traditional ways to go for it. So they were successful, but what happened next, because drugs were optimized for specific combination, for combination with fluoroquinolones, levofloxacin, and so on, target modification became prevalent in clinics. So it's not efflux, which is major mechanism of resistance, but target modification now. So these inhibitors were delayed. So essentially, because the question is difficult, these drugs are specifically targeted for mechanism of resistance, efflux, right? We need to find applications that are specific. Like, for example, acinetobacter, buffer, right? Those where you see perfect efflux only, macrolides and so on. There is an opportunity again, unless while we will be doing this, new mechanisms will emerge and we will be late again. So.
Well, thank you very much. You have a question from Professor Kim Lewis. Helen, great talk, of course. Have you thought about an ideal penetrating compound that would combine properties of inhibitor and penetration avoidance?
So Rempex compounds, right? They are actually ideal in some terms, right? On one hand, they're good permeators. They go across the outer membrane like through butter. And that's one of the examples that outer membrane of Pseudomonas is permeable, right? So then they have antibacterial properties, probably again related to those charges, right? And they're very powerful inhibitors of efflux. So this combination is pretty good. Can we combine different molecules? Of course, right? Again, the combinations will be very much effective. Avoiders, something that we are very much interested to see. So there are avoiders, but there are not so many of them. So there are now, we know some features that make avoiders. Maybe we can expand and actually identify more avoiders and better properties that can be incorporated.
And with that, Professor Zgurskaya, a round of applause. Wonderful talk. Thank you so much. And a round of applause for Professor Fromm and also Professor Brown. Thank you so much for spending your time and preparing a lecture for this conference. I just want to remind everyone to please take a look at the posters and presentations at the website and provide especially the students, comments if you have them. And of course, we're going to have session 2 tomorrow morning. I think it starts at the same time, but please look at the website for the exact times of where you're at to attend the talk. And once again, I want to thank all the presenters and all the conference attendees and the MDPI staff for doing such a great job. And with that, I think we can all say goodbye.
Bye-bye, and thank you, John, as well
right.
for chairing the session.
Right.
Bye-bye.